Assalamualaikum dear students, my name is Dr. Hira and I am course instructor Biochemistry and Genetics too. As all of you know that because of a pandemic which is currently present in on the whole planet, I must say, that is novel coronavirus, we all are in a emergency state where we cannot go outside, where we cannot attend universities and we have to limit our stay at home just in order to avoid the basically the spread of coronavirus. So university management has decided to start online academic courses, online classes, just in order to minimize your loss during the vacations and all. So, as you know that on the last Friday or in the last lecture, we finished our unit of metabolism of carbohydrates, and inshallah, when we will resume our classes in the April, in the very first week, or most probably in the second week of the April, then we will conduct a test of that last unit, which I have taught you. And now we are starting the new chapter, that is the metabolism of protein. And of course, to understand the metabolism and the metabolic reactions of protein, we have to start with the digestion and the absorption of the protein. So before starting with digestion of protein and absorption of amino acids, let's have a very quick review of digestive system components. So the digestive system is basically composed of five parts. The first one is esophagus, second one is stomach, and the other one is the small intestine. Then here is the large intestine. Basically, oral cavity is used for purpose of inhalation, and esophagus is just to pass the food bolus from the oral cavity to the stomach. And then is the stomach, which has two openings, or after the stomach there is small intestine, which is divided into three components: duodenum, jejunum, and ileum. And the last part is large intestine, which is composed of anus and rectum. So this was a very quick review of the parts of the digestive system. After the quick review of digestive system components, let's start with the protein digestion. So what is ingestion? What is digestion? I already have learned you these terms. I already have taught you these terms. Ingestion is food intake. Digestion is breakdown of that food into smaller particle, and absorption is the addition of the digested particles into the blood. And the last part is the metabolism. Metabolism is the digest the absorbed food particles and the absorbed outcomes into the blood and into the cell, where there's those components will be utilized in multiple reactions. So protein digestion is converted into two components. One is gastric part and the other one is the intestinal part. In the gastric part, obviously separate gastric secretions will be released in order to facilitate the digestion, and in the intestinal portion, pancreas and some gastrointestinal secretions, they will be released in order to finish the um, digestive process. So let's start with the presentation. Digestion is the integration of complex nutrients into the simple nutrients which are absorbable from the blood. And as we know that ingestion takes place in the oral cavity, digestion takes place in the stomach and in the small intestine. And most of the nitrogen in the diet is consumed in form of protein, and protein are too large to be absorbed, because always remember when we take up the food which is proteinaceous in nature, then that is present in the polypeptide form, and the purpose of digestion is to convert that polypeptide portion into the very small particles, that is amino acids, so that those amino acids can cross your small intestine wall, and that those amino acids can become the part of your blood. and there they can be recycled or they can be utilized in some metabolic reaction, and through those amino acids we can make multiple products. So dietary proteins are hydrolyzed amino acids by proteolytic enzymes which can be easily absorbed. So now what is a proteolytic enzymes? As the name shows, proteo is related to protein and lytic is broken down, or the enzymes which break down the proteinaceous food or the food which contains the peptide bond. Always remember that the protein has protein is a polypeptide molecule and it has peptide bond which is made up between the amino group and the carboxylic group, and the amino group it contributes hydrogen group, and the carboxylic group it contributes hydroxyl group, and the H from the amino group and the OH from the carboxylic group, they both combine to form water, and the water molecule is released and it is known as the dehydration or the condensation reaction. And when this condensation reaction takes place, two amino acids they combine to form the water, and the water molecule is released, and it is to break down uh, these peptide bonds through their action. So, what are the characteristics of proteolytic enzyme? Their hydrolysis. I already have told you that as they are the digestion of protein, so they may be endopeptidase. That means that the exopeptidase are the enzymes that act on the very terminal bonds of the polypeptide chain, and the endopeptidase are the molecules that they act on the inner bonds of the polypeptide chain. That is why the whole polypeptide chain it is broken down into the small fragments, and when the polypeptide chain will be broken down into the small fragments, then the simple and the free amino acids will be the product. So in the what happens in oral cavity in the digestion of protein, there are no proteolytic enzymes present in the saliva, as we know that in the saliva, the carbohydrate digestion, for carbohydrate digestion, there is a salivary amylase. But always remember that there is no salivary uh, proteolytic enzyme present. So if if I give you an MCQ in the paper, then you must remember that saliva can lead to digestion of lipids and the carbohydrates both, but there is no proteolytic enzyme for the breakdown of the polypeptide chain. So there is no proteolytic enzyme present in the saliva, and the function of saliva is to mix up and to lubricate the food, which makes food more easier to pass from the oral cavity into the stomach. After mastication and the chewing through the teeth, the bolus of the food enters stomach. It is acted upon by gastric juice. Gastric juice is basically the secretion of the which is a bactericidal action of the food particles, and four proteolytic enzymes. And these enzymes are pepsin, renin, gastrin, and gelatinase. As I already have told you, HCl it has a bactericidal action, which is too dilute to hydrolase, but it can cause denucleation of some bonds present in the polypeptide chain or the protein. And it is also required for the activation of inactive proteolytic enzymes secreted by the gastric mucosa, and it has a strong bacteriostatic action. That is the purpose of HCl is to minimize or just and to stop the growth of bacteria. So let's talk about the function of the first proteolytic enzyme, that is pepsin. Pepsin is also released in inactive form, that is pepsinogen, and it is activated by HCl. Pepsin is an endopeptidase, that is, it acts on the inner bonds of the polypeptide chain, and it is active for a peptide bond where amino group is contributed by the aromatic amino acids. What are the aromatic amino acids? I already have taught you in the biochemistry one. These are the amino acid which has a benzene functional group. For example, alanine, tryptophan, and tyrosine. and it has a strong action on milk protein, casein. Always remember that casein is a milk protein which is present in the milk of the cow and in the milk of the human as well. So pepsin, what is the function? What is the basic function of pepsin? It converts protein into peptones and small peptides, and peptone is the soluble form of the polypeptide chain. Afterwards, here comes the renin. Renin is a basically enzyme which is present in infants. Always remember that as an infant, the only the single diet of the infant is renin, is milk. So renin is present in neonates, and it is involved in the curdling of milk. And it is not present in adult gastric juice. Its action is similar to pepsin, and it also acts on the milk protein and is involved in curdling of milk. And gastrecin and gelatinase, the actions are both of them are also it's acid stable, and they are also uh, activated through HCl. Gelatinase acts on gelatin and it converts gelatin protein into short peptide chains. And gastrecin, it also acts on the larger polypeptide chains and convert those large polypeptide chains into small polypeptide chains. Um, this is the overview of secretions, the action of secretions on the protein food. So this is the lining of and the cells of the oil glands are present in the stomach. Three types of cells are present in the stomach: parietal cells, sheep cells, and mucus cells. And these are the places where they are the actions performed by the proteolytic enzymes, and this is the site of release the cells or the glands of the. So, uh, if we revise the previous components, in the oral cavity, no proteolytic enzyme, and in the stomach, there are four proteolytic enzymes: pepsin, renin, gastrecin, and gelatinase, and all of those convert polypeptide chains into small polypeptide chains or into the soluble form of the polypeptide chains. And the enzymes, they are the trypsin, chymotrypsin, elastase, collagenase, and carboxypeptidase. So, what are their actions? What is the action of trypsin? Trypsin is also released in inactive form, that is trypsinogen, and its activation occurs through a hormone, that is enterokinase, which is released from the intestinal wall, the wall of the small intestine, from the mucosa, and it converts basically it acts on milk protein casein. It also converts fibrinogen into the fibrin and proelastase into elastase. What is the action of chymotrypsin? Chymotrypsin, it basically supports the role of trypsin, and it breaks the peptide bonds present between the aromatic amino acid and also work on the milk protein. And the role of carboxypeptidase is converted into two isomers. One is carboxypeptidase A and the other one is the carboxypeptidase B. Carboxypeptidase A has some metals present in the chain in its structure, and it basically breaks the bond. It is a exopeptidase. So it breaks the terminal peptide bonds from the polypeptide chain, and carboxypeptidase elastase and collagenase, as the name is showing, that elastase acts on elastin and collagenase acts on collagen. So this is a very small representation of broken protein into the amino acids, as it's this The diagram is showing that trypsin, chymotrypsin elastase, they lead to formation of these following amino acids: tyrosine, phenylalanine, leucine, arginine, lysine, tryptophan, phenylalanine, tyrosine, methionine, leucine, alanine, glycine, and serine. So we just have a look on this. And afterwards, after the gastric juice, pancreatic juice, there comes a third juice, that is the intestinal juice. And the enzymes present in intestinal juice are enterokinase, aminopeptidase, prolidase, and tripeptidase. Remember that these are the final enzymes which will convert that the small peptide chain into the free amino acids. Enterokinase, it is an enteropeptidase, that means that it will break the inner bonds. Aminopeptidase is an exopeptidase, it will remove the amino acids from the terminal ends of the peptide chain. And the prolidase, the breakdown of small peptide chains into the free amino acids. The oral cavity, it did nothing with the protein or the polypeptide chain. The gastric juice, it divided these some of these polypeptide chains into peptide chains and into the peptones. And the intestinal juice and the pancreatic juice, it will lead to complete breakdown of the peptide bonds and the peptide chains into the heme. So let's see the functions of the remaining intestinal enzymes. Enterokinase, it's an enteropeptidase. It is present in epithelial cells of brush border of duodenal mucosa. Bile salt help in its liberation into the intestinal lumen. Aminopeptidase, it removes the amino acids one by one from amino terminal end of peptide chains. It is also an exopeptidase. It removes proline residues from terminal end of peptide chains. Di and tripeptidase, as the name shows, the di and tripeptidase means that the tripeptidase will act on the polypeptide chains, which has three bonds, and the dipeptidase, it will act on the peptide chain which has two bonds in it. Digestion of di and tripeptidase is brought about by di and tripeptidase is present in brush border membrane of epithelial cells as well as present in a teat of the cell, and tripeptidases convert tripeptidates in tripeptidase tripeptide di sorry, it is tripeptidase convert tripeptide into peptide, and a free amino acid is released, and then dipeptidase convert dipeptides to free amino acid. It will duodenum and jejunum, and from there they will get entry into the interstitia, and through interstitia they will be circulated along with the blood supply, because when the heart pumps, the whole blood supply is mixed up, and all the components, they will be circulated to the required or the target organs or the tissues. So this is how we get free amino acids, and now these free amino acids will get entry into the cell, and there is absorption of amino acids. Proteins are completely digested to consistent amino acids, but some amounts of oligopeptides may remain undigested. The products of digestion are rapidly absorbed. Site of absorption में हम ये बात करेंगे जी के oligopeptides are absorbed from duodenum and the proximal jejunum, while amino acids are absorbed from ileum and the distal jejunum. Always remember that the things that that don't get absorbed from the last portion of the small intestine, they get entry into the large intestine, and in the large intestine all the present material is deposited. And mechanism of absorption, what is the mechanism of absorption of amino acids here? Amino acids, they get absorbed through active transport. Active transport moved against concentration gradient with help of ATP, and D amino acids are absorbed by simple diffusion, and vitamin B6 is involved in the active transfer of amino acids. And energy requiring process ATP is required as a source of energy, and this is the same at ATP that I already have taught you that was produced in form in the process of glycolysis when we were discussing the metabolism of carbohydrates. So a carrier protein is also required independent or independent. That means a facilitated diffusion can also be used for the absorption of amino acids that we have the amino acids they need to cross the intestinal wall, and different carrier proteins are there specific for different amino acids. And um, after the discussion of digestion and absorption portion, this is the food allergies. So all of us know that in a number of persons there is a complaint that whenever I consume egg, whenever I consume an egg, my complaint is that I am having some rashes on my feet or on my legs or on my hands. So that is basically due to food allergies, and food allergies are due to absorption of undigested peptides in the damage in the intestine, which can be presented in form of nausea, vomiting, diarrhea, abdominal cramps, or some skin rashes, runny nose, facial flushing, bronchoconstriction, or some other complaints. So the food allergies they are basically because of absorption of undigested peptides, and this process occurs with help of neurocytokines. So what is the summary? Um, Summary is again no digestion of the protein in the oral cavity, and four enzymes they come in the stomach to break the polypeptide chain into smaller form, and four pancreatic enzymes and five intestinal enzymes they all come and they convert proteolytic they do proteolytic activity, and they convert polypeptide chain into the peptide chains and eventually into the tripeptide and end in the dipeptide, and dipeptide break leads to release of free amino acids. So, guys, this was about the digestion and absorption of amino acids and protein. I tried my best to make it simpler for you. I know that it's this for the first time for you guys, for you to undergo this situation where we cannot interact. Inshallah, when we will be back after the vacations are over, then we can again have a discussion if you want on these topics. Otherwise, you can feel free to ask me any question because I have shared my email address with you, and I'm also present on the Google Classroom where we all are connected through the that media. So you can ask any question if you want to ask. The things are very easier. Start practicing these components in the form of flow sheet that I already have taught you in the digestion of carbohydrates. So if you want any question, then you can pose a question into the Google Classroom, the comment section. Otherwise I can ask you again tomorrow if you have any query. Thank you so much. Thank you.